Hi students, welcome to Smart Locus. In this video we will see the chapter Photosynthesis in Higher Plants, a chapter under plant physiology. In the introduction, NCERT is talking about two experiments, that is about variegated leaf experiment and half leaf experiment. So what is this variegated leaf experiment? Variegated leaf means a leaf which has both green and non-green part. So variegated leaf, if we kept it in sunlight for a few hours and after that if it is tested for the presence of starch, the green part will show positive response, presence of starch. So this experiment proves that green part, that is chlorophyll, is necessary for photosynthesis. So it is kept in sunlight, so sunlight. So light and chlorophyll are necessary for photosynthesis. Second one, half-leaf experiment. Molds half-leaf experiment. A part of leaf is enclosed in a test tube containing KOH, potassium hydroxide soaked cotton, and the other part is exposed. And if this experimental setup is kept in sunlight for a few hours, and after that if it is tested for the presence of starch, the exposed part will show positive response. So this experiment proves that carbon dioxide, KOH, soaked cotton, absorbs carbon dioxide inside the test tube. So carbon dioxide is necessary for photosynthesis. Then let us see few early experiments. First one, contribution of Joseph Priestley. He conducted bell jar experiment by using both mice and burning candle. He kept a burning candle in a closed space and found that after a few minutes, burning candle gets extinguished. And mouse soon gets suffocated. But when he conducted this experiment with a mint plant, animal, that is the mouse, continued to live. Burning candle continues to burn. So from this experiment, he concluded that both the breathing animal and burning candle somehow damage the air. Plants restore to the air whatever the breathing animal and burning candle removes. That was the conclusion of Joseph Priestley. Then later, Jan Ingenhouse, he repeated Joseph Priestley's experiment and found that sunlight is necessary for photosynthesis. And he also proved that during photosynthesis, oxygen is released from green part of the plant. Then comes Sachs, Julius von Sachs, discovered that glucose is the product of photosynthesis which is stored as starch. And then green bodies, that is nothing but chlorophyll present in chloroplast, that is responsible for photosynthesis. Then T.W. Engelmann described the first action spectrum for photosynthesis, that is, the rate of photosynthesis is maximum at blue and red region of the spectrum. He conducted this experiment by using a green algae, that is Cladophora. Cladophora were grown in a suspension of aerobic bacteria. Then comes to Van Niel's experiment. Two contributions. First one is photosynthesis is essentially a light dependent process. And second one, during photosynthesis, carbon dioxide is reduced to carbohydrate. In higher plants, carbon dioxide reduced means hydrogen is added. That hydrogen donor is water. So water splits. As a result, during photosynthesis, oxygen evolution takes place in higher plants. Whereas in lower bacteria, photosynthetic bacteria, like purple bacteria, green sulfur bacteria, H2S is the hydrogen donor. Therefore, in these cases, photosynthesis, the process of photosynthesis does not involve the evolution of oxygen. Next, about site of photosynthesis. Where does photosynthesis takes place in plants? In plants, or leaf is the organ specialized for photosynthesis. Then within leaf, mesophyll Mesophyll is the tissue where chloroplast is present. Chloroplast is the organelle for photosynthesis. Is there any plant in which stem or root perform photosynthesis? Yes. Root perform photosynthesis in Trapattinospora. We call it as assimilatory root. Stem perform photosynthesis modified for carrying out photosynthesis in Euphorbia opentia. Then, alignment of chloroplast. Usually, chloroplast is aligned along the walls of mesophyll to get optimum quantity of light. Then, these chloroplasts are aligned parallel to the walls of mesophyll, that is parallel to incident light. Parallel to incident light during high light conditions. This is to protect chloroplast from photooxidation. Then, chloroplasts are aligned perpendicular to incident light. Perpendicular to incident light during low light conditions to get that optimum light. Within the chloroplast, there is a membranous system. Membranous system comprises grana formed of thylakoids, then stroma lamellae, membranous tubules that connects adjacent grana, then fluid stroma. So grana, stroma lamella, fluid stroma together constitute membranous system, which is responsible for trapping sunlight and synthesis of ATP and NADPH. These two are nothing but assimilatory powers required for the next step, that is dark reaction to occur. So this reaction is known as light reaction. So membranous system is responsible for light reaction of photosynthesis. Then stroma contains enzymes for dark reaction. Starch is the ultimate product. Glucose is the immediate product, which will be stored as starch. So see, light reaction, dark reaction. Light reaction, membranous system. Dark reaction, stroma. Then next about pigments. Pigments in photosynthesis. Chlorophyll A is the major pigment. Blue-green in color. Chlorophyll B, yellow-green in color. Xanthophyll, yellow. Carotenoid, yellow to orange-red. Then absorption spectrum of chlorophyll A shows that chlorophyll A absorbs maximum light at two wavelengths, that is blue and red. And absorption spectrum of chlorophyll A coincides with the action spectrum also, that is the rate of photosynthesis is maximum at blue and red region. Then other pigments like accessory pigments. Accessory pigments means chlorophyll B, carotenoids, xanthophyll. They absorb light of other wavelength, different wavelength, and transfer it to chlorophyll A, which is the reaction center. That is one of the function of accessory pigment. The other function, they protect chlorophyll A from photooxidation. That is light-induced oxidation of chlorophyll. Light reaction. That is the photochemical phase of photosynthesis. Light reaction or photochemical phase involves absorption of light, then splitting of water, then release of oxygen and synthesis of ATP and NADPH. The next point, what is LHC? Light harvesting complex. The pigments are organized into LHC, light harvesting complex. Light harvesting complex composed of pigments and proteins. This light harvesting complex is organized into photosystems named photosystem 1 and photosystem 2. Both the photosystems, whether it is 1 or 2, each photosystem has a reaction center and antenna complex. Reaction center consists of chlorophyll A molecule. The antenna complex consists of other pigments. In photosystem 1, the reaction center is known as P700. That is, chlorophyll A molecule has an absorption peak at 700 nanometer. So, that is the reaction center in PS1, photosystem 1. In PS2, it is P680. Absorption peak at 680 nanometer. So, P700 stands for photosystem 1, P680 stands for photosystem 2. Both of the photosystem has a reaction center and antenna complex. Next, about electron transport. The two types of electron transport are non-cyclic electron transport, also known as non-cyclic photophosphorylation, and cyclic electron transport, cyclic photophosphorylation. First, let us compare these two. Grana, non-cyclic electron transport takes place in grana, that is thylakoid membrane. Then cyclic electron transport, stroma lamella is the site where only cyclic takes place. Stroma lamella has no PS2, so no NADP reductase enzyme, so it has only PS1, so only cyclic electron transport takes place. Then non-cyclic, both PS1 and PS2 takes part, here only PS1 takes part. Both ATP and NADPH are synthesized, here only ATP is synthesized. Then photolysis takes place during non-cyclic, which involves the release of splitting of water and evolution of oxygen. But in cyclic, there is no photolysis, non-cyclic electron transport. PS2, photosystem 2, gets excited when light falls on photosystem 2. And the ejected electrons are accepted by a primary electron acceptor, that is phyophytin. And from phyophytin, these electrons are then accepted by the next carrier, that is plastoquinone. Plastoquinone is actually a hydrogen carrier. And from plastoquinone, these electrons are then received by cytochrome B6F complex. And from cytochrome B6F complex, the next carrier, that is plastocyanin, receives electrons. And plastocyanin transfers this electron to photosystem 1. Simultaneously, photosystem 1 also gets excited when light falls. And the released electrons are accepted by primary electron acceptor. From where that electron goes to Ferredoxin, and from Ferredoxin, these electrons are ultimately accepted by NADP+, which in turn gets reduced into NADPH plus H+. Photosystem 2 shows the deficiency of two electrons, that is the two electrons rejected from photosystem 2 are not returned. And associated with photosystem 2, there is a complex known as water splitting complex, which splits water in presence of light, that is known as photolysis. And this photolysis releases protons, electrons and oxygen. This electron goes to photosystem 2, the ultimate donor of electron in this non-cyclic electron transport system. The ultimate donor is water only. The ultimate acceptor, the final acceptor here is NADP+. And the whole scheme of reaction, that is known as Z scheme. And here, based on redox potential gradient, the electron flow is first uphill. That is from lower redox potential to higher redox potential. From photosystem 2 to phyophytin, it is uphill. And from phyophytin to photosystem 1, plastocyanin and photosystem 1, it is downhill. That is higher redox potential to lower redox potential. And from photosystem 1 to primary electron acceptor, again it is uphill. And from primary electron acceptor to NADP+, again it is downhill. So see, it is uphill, downhill, uphill, downhill. Photolysis, the water splitting complex, is associated with photosystem 2, which is located on inner surface of thylakoid membrane. As a result, protons formed during photolysis are released into thylakoid lumen, that is thylakoid cavity. And during electron transport, non-cyclic electron transport, from photosystem 2 to photosystem 1, ATP synthesis takes place, that is phosphorylation takes place. So the two products formed during non-cyclic are ATP, then NADPH plus H+. Oxygen is also released. The mechanism of ATP synthesis during this electron transport is explained by chemiosmotic hypothesis. Chemiosmotic hypothesis. This hypothesis was proposed by Peter Mitchell. See, according to this hypothesis, an proton gradient is necessary for ATP synthesis. That is, the breakdown of proton gradient gives energy for ATP synthesis. Requirement for chemiosmotic synthesis of ATP are a membrane, here thylakoid membrane, proton pump, proton gradient, and ATP synthase enzyme. Proton gradient develops across thylakoid membrane between stroma and thylakoid lumen. Between stroma, see here this is thylakoid. If we assume this one as thylakoid, inside thylakoid lumen, outside stroma. That is the matrix of chloroplast. Then, let's see how this proton gradient build up. Photolysis releases protons into the lumen, which increases proton concentration in the lumen. Second point, see, during electron transport across the membrane, see electron transport from PS2, then through electron transport system, PS1. So during electron transport across the membrane, proton transport takes place from stroma to lumen. See, actually this is the proton pump. How it works? Actually, phyophytin, the primary acceptor of electron from PS2, transfer its electron to plastoquinone, which is actually a hydrogen carrier. So it takes H plus from the stroma and then it forms PQH2. From this plastoquinone, cytochrome, the next immediate carrier which is located towards the inner side of the membrane, thylakoid membrane, receives only electrons because it is only an electron carrier. As a result, protons are released into lumen. So this is the proton pump, plastoquinone, cytochrome. Plastoquinone is a hydrogen carrier. Cytochrome is only an electron carrier. So this is the proton pump which works during electron transport. So second reason, which increases proton concentration in the lumen. Third point, see from PS1, Ferredoxin, electron goes to Ferredoxin, then FNR, that is Ferredoxin, NADP, Oxido, Reductase. This enzyme, located on the outer surface of thylakoid membrane, receives electrons from electron transport system from PS1. But it takes H plus from the stroma to reduce NADP plus into NADPH. That means this enzyme activity decreases proton concentration in the stroma. So these are the three reasons responsible for creating a proton gradient across thylakoid membrane. That is, more protons in the thylakoid lumen. The enzyme ATP synthase is responsible for ATP synthesis. Actually, this enzyme has two components in the structure. See, CFO, then CF1. These are the two components. CFO is actually an integral membrane protein located within thylakoid membrane. It acts as a proton channel. It allows facilitated diffusion of protons across the membrane. CF1 is the catalytic part of the enzyme ATP synthase. When proton flows from thylakoid lumen to stroma, first it will pass through the FO part. Then when it passes through the F1 part, it brings a conformational change in the enzyme. As a result, ADP and inorganic phosphate present in the stroma combines to form ATP. ATP synthesis takes place in the stroma. Actually, this diagram is given in NCERT 2020-21 edition. Here, since light energy is responsible for creating the proton gradient, the mechanism of ATP synthesis is known as photophosphorylation, cyclic electron transport. Only PS1, photosystem 1 is functional. And here first, when light falls on photosystem 1 gets excited, electrons are accepted by primary acceptor, and from primary acceptor, this electron passes through a series of electron carriers collectively called as ETS, electron transport system, and finally that electron returns to photosystem 1. During this electron transport, ADP combines with inorganic phosphate and ATP synthesis takes place. That is why it is known as cyclic photophosphorylation.